When you're looking at diabetes research, there are many differences between mouse and human, which emphasises the importance of needing to use human models for human disease.
I think the big thing with our research is that we're working in an entirely humanised system. So most of the work that goes on in this type of field is done with animal cells in culture. The pattern of genes which are switched on is sometimes very different in mouse cells, for example, compared with human cells. So actually, if we had been doing this research in mouse, we would never have found what we found.
Hi, I'm Nikki. I'm a PhD student working at the University of Exeter Medical School.
I am Professor Lorna Harries. I'm an Associate Professor in Molecular Genetics at the University of Exeter Medical School. We work on diabetes. In particular, what happens to the cells in your pancreas that make insulin when you have diabetes? So people used to think that during the course of diabetes, as you sort of go through your life with diabetes, that the cells in your pancreas which make insulin, which are called beta cells, they died. What our research has shown is that actually those cells don't die. They just turn into a different kind of cell, and that other kind of cell doesn't make insulin. And it raises the possibility that we might be able to persuade them to turn back into beta cells in the future.
We're using a human beta cell line and also tissue sections from patients with type 1 and type 2 diabetes, as well as people without those conditions who have also donated tissue sections. These are post-mortem patients who have died, so they're very precious samples. What we're using those samples for is to look at the effects of diabetes on beta cells and what it does to the beta cells. So I'm basically interested in what is lifestyle doing to those beta cells? How is it stopping them from working, and is there something that we can do about that?
So the other thing that our research has found is that if you can control the environment that your cells are living in, either by keeping your weight under control or by keeping your blood sugars equal, you can protect the cells in your pancreas from turning into other types of cells, and you can maintain the ability to secrete your own insulin.
You might have heard stories in the media of people saying that they've been able to reverse their type 2 diabetes. And what we think is this might be the mechanism by which that is happening. From what we've seen from the beta cell model that we're looking at, is the cells are telling us that it is possible to reverse it. What we can tell people is that yes, if they are able to modify their lifestyles, then they have potential hope there for going into remission with their type 2 diabetes.
We could have done these experiments in mouse cells, we could have done them an awful lot quicker. We wouldn't have found what we found, but the chemicals and things that we're using to do those experiments are about 10 times more expensive if you're looking at human systems. Because the methods are not out there, we had to make the method. Hopefully now we've made that method and other scientists will now come and employ it and use our system for their work.
If you care about removing the need for the use of animals in research, so if you feel strongly about it, then it needs to be funded because the alternatives are more expensive.